amazing students, and welcome to my first shot at teaching online. I have no idea how to do this. I hope it works, and I hope that you're able to learn from it. Know that I miss all of you, and I hope that you will soon be back here enjoying science. But for now, we're going to start with a review of Lesson 10.3, DNA and Genetics. Most of you already have seen a lot of this, but most of you have been off for a week and have forgotten a lot of this. So, to begin with, here's the vocabulary. We're looking at DNA, which of course stands for deoxyribonucleic acid, nucleotide, replication, RNA, transcription, translation, mutation, and genetic engineering. This first lesson hopefully will only be about 10 minutes and will be a review of what you should remember learning right here in this classroom. Our essential questions are: what is deoxyribonucleic acid? What is the role of ribonucleic acid in protein production? Remember, proteins are the key to everything. And how do changes in the sequence of DNA affect traits? We did this code in class together, and hopefully you remember the answer is DNA provides the chemical code for an organism's genetic material. And that really is the heart and key to what we're learning about here. We looked at the definition for genes and DNA. DNA is an organism's genetic material. A gene is a segment of the DNA. So here's our chromosome, and here's our long double helix strand of DNA, and chunks of this are the genes for the different characteristics that make you you, and make the nifty proteins and things that you need. So we solved this in class. We wanted to look at how our DNA was made, and we saw that the sides are made out of sugar and phosphate groups. On our model of DNA of science here, you can see the sugar and phosphate is this purple and reddish color. Then the teeth, or the rungs of the ladder, are called the nitrogen bases. Remember, they're in the four-letter code of A, T, C, and G: adenosine, thymine, guanine, and cytosine. And together with the phosphate and sugar and the nitrogen base, we get a nucleotide. So those are some of the fancy words that you'll be putting on your concept map, that you'll be taking a picture of and uploading to Google Classroom soon. Then we looked at how big things are. We wanted to know from biggest to smallest. We decided that the largest thing was the cell. Inside the cell, there's the nucleus with the nucleolus. That's where the DNA is. Then, just a little bit of foreshadowing for a future lesson. See this weird looking stuff here? That's one of my favorite words in science. It's called Endoplasmic Reticulum. This is the Endoplasmic Reticulum. And the Endoplasmic Reticulum is kind of a pathway for a clever thing called messenger RNA to travel through. And all these little spots, this thing doesn't have smallpox, all these little spots are ribosomes. And ribosomes are little factories for making proteins. So that'll be important later. Inside the nucleus are the chromosomes, and the chromosomes are the second biggest thing. Then the coiled coils of DNA are our third biggest, and then finally the nitrogen bases. We looked at how long a strand of DNA is and decided on about 6 feet or 2 meters. About how far you should be socially distant, so you can remember. How long is a DNA strand? About how far away you should be standing from anybody else. Chromosomes, genes, and DNA bases, we have about 25,000 genes on our chromosomes. It takes 25,000 instructions to make you. and to make me. These instructions are on these genes that are on these coiled coils of DNA. There's 23 pairs, of course, in humans, 46 total, and 3 billion base pairs. That's the code to make a human being. We have 10 to the 14th cells in our body, and we figured out that that was about 100 trillion cells. Then we've had the question, how can the super long strand fit in this tiny little space? And the secret is the histones. The DNA wraps twice around each histone, and then you have it all coiled up, clicking together, kind of like Lego blocks, building your chromosomes. We look at how DNA replicates. It's important for DNA to make copies of itself. If it can't make copies of itself, it can't make new cells. You can't grow, nor can you heal, because the only way that we heal is by making new cells. The way that we grow is by making new cells. To make new cells, the DNA has to copy itself. So this is replication. The process of copying a DNA molecule to make another DNA molecule is called replication. You can follow the steps of DNA replication in the image. Each strand separates in many places, exposing individual bases. Then nucleotides are added to each exposed base. This produces 2 identical strands of DNA. We'll see the picture for that in a minute. But imagine this strand of DNA splitting down the middle, but not fully separating, and then other nucleotides being attached to it, so that eventually you get 2 complete new strands of DNA. Then there's RNA. This is critical in making proteins. RNA is a type of nucleic acid that carries the code for making proteins from the nucleus to the cytoplasm. RNA also carries amino acids around inside a cell and forms a part of ribosomes. Remember, I told you those ribosomes are going to be a big deal coming up later. So really important to know about RNA, which helps use the DNA to build all the stuff that is you and all the stuff that is every other living thing. And finally, transcription. The process of making messenger RNA from DNA is called transcription. The image shows how messenger RNA is transcribed from DNA. I've used a lot of big words. Let me try to explain what I mean here. Well, first we have to solve this little thing, which we already did in class. Describe the shape of a DNA molecule. Yeah, you're all saying twisted ladder, aren't you? Some of you sharp people are saying double helix. It's a double helix or twisted ladder. Identify the 4 nitrogen bases. They're A, T, C, and G. Adenine, thymine, cytosine, and guanine. And A always bonds to T. A always bonds to T. And C always bonds to G. There's a little quiz I put on Google Classroom that uses that fact to see if you can read DNA. I hope you find it kind of fun and easy. Okay, so here's replication. To make a copy, the DNA splits down the center. Do you remember what kind of bonds I told you held the nucleotides together? Yeah, you're all saying hydrogen bonds, aren't you? You're brilliant. I like you. Okay, the DNA strand separates, then the new nucleotides are brought in, and 2 new strands of DNA are formed. And that's the first step in mitosis, or cell division, in separating and making 2 new cells, and healing, or growing, or building proteins. Then here is the transcription thing. In this case, we're not trying to make a copy of the DNA. In this case, the DNA splits just enough to release the gene for the code for the messenger RNA. This strand of messenger RNA is made as these new nucleotides click in, and this strand is on its way through the endoplasmic reticulum out to the ribosomes to help make proteins for you and me. Alright, so we looked at this. A DNA strand separates and nitrogen bases are exposed, so the new ones can click on. Nucleotides form new nitrogen base pairs, and the result is 2 identical strands of DNA are produced. Then we get down to the RNA and describe 2 functions. RNA is a type of nucleic acid that carries protein-making codes. It also forms a part of ribosomes. You could pause the video and write this down in your book. If your book is still here, you can call and make an appointment to come pick it up, or send me an email. Alright, the next thing we're going to look at is translation. Turns out there's 3 types of RNA, and that is new information, so we'll take a look at this in the next lesson. Thanks for watching. Stay healthy. This is Mr. C.